I'm just recording using my PC. Hopefully it's working like last time. Okay, so last Tuesday we've looked at some development pipelines, and then we also looked at what you can do if the drug that you design actually doesn't work. Okay, doesn't work as planned. It's not that it doesn't work at all, but it doesn't work as planned. Okay, so number one, we've looked at delivery methods. We talked a little bit about it, differences, the different types of delivery methods, and we did not really look at what are the advantages and disadvantages because it's too many, and it's not part of the learning program. And then we also look at prodrugs, and we stop here. We will look at what product is. So prodrug is basically inactive form of that particular drug. So in this case, that I've shown last week or last Tuesday was L-Dopa. So L-Dopa is a prodrug for dopamine. So now I remember what is the active ingredient in our brain. Okay, so how does it work? So you don't have this slide, but I'm just gonna show you why L-Dopa is considered as a prodrug. Okay, so over here, this is L-Dopa, okay, and this is dopamine. So what our brain needs, or what of the chemical receptor in our brain is dopamine. So as we grow older, we have issues in synthesizing dopamine. Not everyone, but some will have some degradation in the genetic markings and whatnot. So the body will not be able to produce enough dopamine. So thus the disease that is related to dopamine is Alzheimer. So what researchers and pharmaceutical companies have developed is what they call as L-Dopa. It is part of a prodrug. Okay? So L-Dopa on its own, in the presence of decarboxylase or methyltransferase, will become either multidopa or dopamine. But this dopamine needs to be in the brain for it to function. And dopamine alone, it does not penetrate into our blood, we call it as blood-brain barrier. So there's a distinguished barrier that kind of like isolate our brain from the other system of our body. It's similar in in a baby, in a fetus, in the womb. Okay, they have their own system, still share with the mom, but it is nonetheless a separate system. So dopamine on its own, it cannot passes through blood-brain barrier very easily. Okay, and similarly, methyldopa also does not penetrate and passes through into the brain very easily. But L-Dopa, the inactive form, does that very readily. Okay, so this is the form of a drug that is being developed, passes through the blood-brain barrier, enters the brain, and then in the presence of a different enzyme, okay, and over here there's still decarboxylase. Okay, so different enzyme either kind of like trying to get rid of the prodrug, okay, either forming this and this, or some of them will also convert it into dopamine and thus being used by the brain. Okay, so it's not directly involved. If you just develop dopamine, okay, if you are the first person who developed dopamine a long time ago, you test it in say in a cell culture, for example, and it helps the brain cell to do whatever it needs to do. But then when you test it in human, it doesn't work. Okay, but instead of just throwing away that idea, you can actually convert it to something else, and that something else, a prodrug, actually works as what is intended. Okay, so that's an example, and that's the reference if you want to read more about this. Okay, the third option is optimizing drugs. We also look at that in a little bit, looking at all these things, and I think we stopped at application combinatorial. We did that. Okay, we've stopped at here. So statewise chemistry. So in doing such a relationship activity, this one is the SAR. So you have three approaches. We've looked at approach number one, developing a library using combinatorial chemistry. The second approach is more on stepwise chemistry. So what does it mean is that you do one by one. So instead of like combinatorial chemistry, you just do randomly, getting all your samples and then you test all your samples. But this one, you do it one by one, stepwise. Meaning that, say perhaps you synthesize a molecule, okay, and then that molecule has a certain activity. So nowadays, pharmaceutical companies looks at a very low dosage, meaning that they want the drug to be as efficient, as effective as possible. So you go down until a nanomolar concentration. So say for example you are developing a drug, and then from that drug you manage to get a micromolar scale. Okay, so micromolar, still not too bad. It can be what we call as a hit or a lead drug based on our previous lecture. Okay, so from hit and lead, we then modify one by one and then do another experiment. So you modify one, for example, you add in a methyl, changing a hydroxyl into a methyl group, or adding a methyl, not converting to methyl. You can remove it, but yeah. So there's a lot of modification that you can do, and then from that modification, you go back again to the lab and see if your modification actually promotes or deactivate a certain activity of that particular drug. Okay, so a good example, again, this I've shown this drug previously, which is HIV-1 protease inhibitor. That's a structure. Okay, and this is another similar type of drug. So this one is a substitution based on that structure. Okay, so the R's and whatnot, so you can go back, and I think your lecture is slightly different than what I'm showing you. But this is another HIV inhibitor. So now because you guys know about Pymol, you can actually go to Pymol and key in this 1QDS. You should be able to get this structure. And if you look at this structure over here, okay, somewhere in the middle there, the structure actually has similar ring. The basic ring is similar, but the side arms are different. Okay, so this is just one example on doing stepwise chemistry, and this drug, I'm not sure whether it's marketed or not, but because this one is from a research paper, was shown to be under nanomolar concentration. So the activity is at nanomolar scale, which is very good. But of course, it's not part of the original drug. But nonetheless, the skeleton are similar, and this is how you can do in your drug design assignment. So you design something, or perhaps if you look at your proteins, right? So you have your own protein target. The protein target might have a drug that has already been tested that binds to that particular protein. So you can use that as a starting point for your modification. You can do that. It's fine, as long as you fit on the criteria, which is either your group is doing a protein peptide base, then the drug that you need to test is peptide base. If it's a lipid, then you can choose steroids and other stuff. Okay, so as long as you fit all the criteria that I requested, you can either find a drug as a starting point, or you can create on your own. Either way is fine. Okay. Alright, so the third one is a computational chemistry. So a different terminology for SAR using computational is what we call as qualitative SAR or QSAR. So if you read some articles and they mention about QSAR, so it's more or less similar to the SAR that you guys are learning now. Okay, the difference is QSAR solely focusing on computational work and not like the old school. So the old school ones, the combinatorial chemistry or stepwise chemistry, those are what we call as SAR. Okay, but the computational ones, they have slightly different name. It's the same thing, but you know, whatever they just want to name it differently. So just use that. Okay, so what it does is you use computational theoretical or computational prowess and modify your molecule in silico. So this is what you guys are doing. Okay, so advantage is modern computation architecture. You have nowadays even your laptop or even your smartphone is way faster. Have you had any class with Prof. Sharifuddin? So did he mention about supercomputer? Yes. He normally says that like the phone nowadays is way faster than the supercomputer that he used way back when he first started learning UM. Okay, so that is the advantage of using computational chemistry nowadays. So you have a lot of computational power. Even your laptop, your iPad and whatnot have more computational power than long time ago. Okay, And on top of that, we are going into our IR 4.0. There's a lot of automation. Okay, so your assignment, if you know what you want to do, you can actually automate it and let the computer finish all the work instead of you doing it on your own. Okay, but this is part of the learning, um, part of your learning. Therefore, I just ask you to do one by one, stepwise. But once you know how to do it, you can actually automate. And 5 molecules are actually very easy to do. You can actually finish all the docking within 2 days. So one day you can just spend and create like 100 or even 200 molecules, and then just let the machine runs for you automatically without clicking anything. So yeah, but if you want, you can search for it. It's there. But otherwise, you can just do it from one by one. Okay. So this is one example of what I did in terms of competition chemistry. So we did, this is just a 4 library, one of the 4 libraries, 4 peptides that we designed that has the best affinity. So even if you look at that, the energy is about 97. So in your own design, you might get a better finding. So it's fine, you can use that. And what is different between what you are doing and what this paper that I've published is that this one is not just looking at molecular docking, we are also looking at molecular dynamics. So dynamics, when you are doing docking, normally you're just looking at rotation of bonds, angle of the bonds, and then a little bit on the interaction between the molecule and your protein target or receptor. Okay? But molecular dynamics is looking at the movement of the whole thing, so including solvents, interaction between solvents. So even if you have a very good docking structure, but if you go into molecular dynamics, that docking structure might not be as good as what is in real life. Okay, because towards the end of the day, when you are doing your docking, remember, as part of the process preparing all the receptors and your ligands is to remove all water. Remember, when you are doing PIMO, you're going to remove all zeros. Okay, so those are all waters. So you remove all water. Therefore, when you're doing docking, the calculation does not involve calculating whether water has an influence. But as you will learn in subject, I can't remember. I also taught a different subject. So water is actually part of an important entity in forming structure, in forming interaction. So if you remove water in your docking, it might show that it's good, but in real life it might be not as good. Okay. So that's molecular dynamics. And then what we do is, after that, is we take whatever results we get, the best results from the docking and the molecular dynamics, we synthesize the peptide, and then we test it and see if the peptide actually works as intended. Okay. So this one, SID001, you can see there's a reduction in about 40%. Okay, and it shows this is the best. Okay, so this is a correlation, just to show you guys the correlation between your docking and in real life experiment. So this one is using cells and using dengue virus. So we are actually looking at whether the drug that we designed in silico works as intended. Okay, so this is just to show you lah. Okay, and number 4 is drug formulation. So drug formulation, there's a few terms that you might want to know here. The first one is what we call as API, or active pharmaceutical ingredient. So it is basically the drug itself, the drug that does all the function against your target. Okay, so that's what we call as API. And the second term that you need to understand is excipients. So what are excipients? Excipients are basically additive to the drug. So you design the drug, but there's no one drug in this world that you designed it, you synthesize it, and then you straight away consume it. Okay, so all drugs are being formulated in some way, one way or another. Again, according to whether you're gonna deliver it, whether route of administration, whether there's a few things that we're gonna touch about that. So we're gonna skip that. Okay, so excipients also may perform a variety of functional roles in the pharmaceutical products. We're going to look at a few examples after this. Okay? And in major cases, excipients have limited, if any, pharmacological activity. So pharmacological activity is what you want API to have, okay, because that is the active ingredient. But nonetheless, sometimes the excipients is also added, and they do have a certain kind of like effects on the receptor target. Okay, one good example is ethylparaben. It is an excipient, that is the chemical structure, and benzocaine, which is the active ingredient, and that's the structure. You can see that part of it are very similar. Okay, but nonetheless, when this drug is being sold, the active ingredient is always says that benzocaine, which if I'm not mistaken is a cough drug. Okay, if I'm not mistaken, I may be mistaken, but if I'm not mistaken, and the excipients, one of them, of course not just one, so there are a lot of other excipients, and one of them is ethylparaben. Okay, so what are the functions of excipients? So there are a few purposes, as you see in the notes. So number one is to promote alternative path for drug delivery. For example, if the drug that you design have a different function or different each target. So the active ingredient might be similar. For example, we take Panadol. Okay, so paracetamol. Paracetamol is good for, not good as in you can just consume it, but it's active for a wide range of age. Nonetheless, a pill version might not be suitable for children. Okay, because children might have problems swallowing, or if they are still babies, you don't want to just chuck it in. So what you do is you change the formulation. So the active ingredient is still the same, but you change the excipients. Okay. And another example for male and females. So you can see Panadols, for example. Nowadays they have menstrual Panadol, and they have a soluble, they have a normal ones. Okay, so in terms of the differences in sex, the menstrual Panadol have extra excipients. They have antiplasmodic excipients. So this one is kind of like an antispasm. So paracetamol itself is a kind of like a painkiller, but the anti-spasmodic excipients is when you have some gugut. Okay, so for the ladies, so that's why this Panadol for guys, do not purchase it. Okay, it's not useful for you. So I'm not sure what will happen, but yeah, this one is specific for ladies. Okay, the second function is to enhance efficacy of a drug. Okay, this one can be related to increasing bioavailability, meaning that how fast the drug, the active part of the drug enters your body and have an effect. Okay, a good example for this one is when you see a Panadol, Actifast, or Optizorb. So what are the differences between normal Panadol, Optizorb, and Actifast? Okay, so it's basically the formulation. If you read, for example, the Actifast, they have about 200 milligrams more sodium as compared to the normal ones, and this sodium helps in our body absorbs the paracetamol into the body. Okay, methods and how it actually does that, you need to actually go and read the notes. I was thinking of going into it in very detail, but then there's too many excipients, so I'm just gonna skip that one. Okay, so just showing you an example. Over here we have regular Panadol, Actifast, regular Panadol again, either in a fat state or fasted state, and you can see there are differences. Okay, so in fat state, meaning that you eat first, you have a meal before you consume the drug. Okay, fasted meaning that you consume it before you actually have a meal. If you look at here, Actifast, just look at Actifast regular. Okay, in the fasted state, you can see Actifast as what is designed for being absorbed in the body very quickly as compared to the normal ones. Okay. Alright. Can anyone give me an idea? Why do they normally test? Just a rough idea. Why do they normally test between fat state and fasted state, and before and after meal? What is the important? Why does there any differences? When you're a kid kan, normally doktor cakap ambil sebelum makan, atau ambil selepas makan. So why is it important? Okay, interaction is Could be one. Yes. Any other suggestion, opinions? What? Yang online pun boleh cakap online ni. Online ni biasanya kalau saya nak bagi nama kan. Mari kita tengok. Ricky, are you here? Okay, why do you think there's a difference between if you take a drug before or after a meal?
Because if we eat food, the chemicals will affect the function of the drugs, effectiveness.
Why? Like, or perhaps, why do you think, or how do you think it will affect on the effectiveness of a drug?
I don't know, blocking the body from drugs.
Okay, so that is actually quite true. So food that you intake either blocks, but not blocking the enzyme per se, but some, thank you Ricky, so some drugs that are being, so some drugs, okay, uses the same enzyme or receptor or transporter than compared to the food that we intake, okay, the food that we consume. So that's why some drugs are formulated to contain, for example, glucose. Okay, because you know when you eat, for example, nasi and whatnot, there's a lot of carbohydrate, there's a lot of glucose. So there is a glucose transporter in our body, in our stomach, in our intestine, not stomach, in our intestine. So this is where you know, if the drug is either being conjugated together with the glucose, will help the drug to enter the body via a different pathway. So they can have a shared enzyme or shared transporter, or they can have an individual transporter, or perhaps they can have what do you call it? I don't know what my head, the terminology. But anyhow, okay, so and they might have a certain side effects. For example, you know that drugs that being consumed will be metabolized in our body, in our liver. Okay, so there are some things that you cannot eat because that thing might be metabolized in our liver. For example, this one is especially for the Westerners where they drink a lot of wine, okay, and a lot of chemical components in wine are being metabolized in the liver. So that's why we can see a lot of drugs that mention that don't drink before consume, or don't drink, as in not drink water, but drink wine, okay, especially, or drink beer, because there's a lot of chemicals in wine and beer that is being metabolized in our liver, and then that enzyme that metabolizes the components in wine might also be used to metabolize the drug. Okay, so say for example, if you consume a drug and then the drug concentration in your body goes up to 100%. Okay, so just imagine lah. Okay, so in the liver, it will degrade the, either oxidize or do something else to the drug, so that as time passes, the drug concentration lowers. Okay, so imagine if you drink or you eat something that uses the same enzyme that does the catalysis for the drug. So instead of the drug, because I was drawing like this, so I forgot that people online cannot see it. So imagine this is what will happen on the concentration of the drug, okay, over time. So it will go down due to metabolism by a certain enzyme. So just say it's an enzyme A, okay, for example. And this is what we normally know, and this is how doctor prescribes drug. So doctor says, okay, based on your condition, normally you will metabolize the drug within 8 hours. Okay, so that's why, for example, paracetamol says that you do not consume paracetamol more than 500 milligrams in 8 hours. Okay, so this is the reason, because after a certain point, the concentration of the drug will lowers. Okay, and once it goes low like this, the doctor might say, okay, take another pill after 4 hours. So what will happen is the concentration will go back again, so that it will have a physiological effect, and then it will go down again. Okay, so this is kind of like the cycle between time points when you are taking a drug. But what if you eat something or you drink something, and then that something actually utilizes the same enzyme. Okay, so what would happen is the concentration of the drug maintains for a longer period. So say for example, after these hours, 8 hours for example, and then you follow what your doctor says, take after 8 hours. So what will happen is the concentration of drug will go even higher. And why is this important? Why is this dangerous? It's because everything that we eat has a toxicity. It's a toxic to our body. So if you go a certain threshold, then it becomes toxic. So that's why it's very important for us to go back and see whether the drug must be taken before or after meal. Jauh pula cerita. Okay, the third one, I haven't touched on that. So the third one is you need to formulate or add in or some excipients to increase drug solubility or perhaps increase the suspension of the drug in the vial itself. For example, okay, in a cancer drug called toxoterone, they are using ethanol and surfactant. So there are only two excipients in that, ethanol and surfactant, which is a polysorbate. So ethanol is to both of these has the same function, which is to as a stabilizer for the drug. But ethanol, on the other hand, also can act as a antimicrobial. Okay, high concentration of ethanol becomes toxic to bacteria. So the drug that you formulate can now live happily for a long time on the shelf before it's actually being used. Okay, but for a drug like this, you can say surfactant. Surfactant is like a soap. If you drink it, then it's very toxic. Similarly, ethanol at high concentration is very toxic. So before you actually, like for example, for this specific drug, before you actually deliver it to a patient, you need to dilute it. Okay? Dilute it in normally special water saline before it's being IV into the body. Okay? Another example is liposome. Liposome also a lipid membrane is also used. Liposome plus cholesterol and whatnot is being used as a formulation as a stabilizer for drug. Okay? A good example for this is which vaccine? I can't remember. I think Pfizer and AstraZeneca has more or less the same constituents. So a good example in terms of increasing stability is Pfizer and AstraZeneca drug vaccines. Okay. The fourth one, why do you need to have excipients, is to ensure the drug are safe. Okay. So what does it mean by that is that sodium chloride, for example, you need it to adjust the tonicity. So we do know that our body has its own osmotic pressure that it needs to be and whatnot. Okay? So some drugs are highly toxic or requires something else to kind of like adjust the tonicity, the content, the water content. Therefore, sodium chloride is added. Okay? So that's one example. Second example is to make sure that the drugs are safe by adding preservative, or in terms of surfactants, for example, or ethanol, the ones that I give you previously, to prevent hypobacterial growth. Okay. The fifth one is a metal chelator, for example, EDTA or EDTA derivative. So why is to make sure that they capture all metals that can catalyze oxidation. Okay, so it becomes an antioxidant. We do have excipients function as a targeting. For example, if the drug is being formulated with a liposome, so liposome, certain types of liposome is a very generic term. So there's a lot of types of liposomes. Okay, so this liposome monomer can actually has a certain function in terms of targeting. Okay, and also VLP, which is virus-like particle. So say for example, if you are trying to develop a cough or flu drug, you might want to use something that is similar to a flu virus. Okay, so that it will target just on the lining of your breathing or your lungs, and it doesn't really go to other places. And there are many other functions, for example, just to change the color. Okay, why? Especially because kids, for example, they prefer colors like red instead of blue or green, because green is normally associated with either grass or plants, or vegetables, right? So if a drug formulation is green in color, they might refuse to actually take it. But if it's green, or if it's their favorite color, red, for example, or blue, so they might actually help in kind of like promoting the kids to consume it very very readily. Okay. Other functions are also stabilizers or pH adjuster. Okay. So these are other examples of drug formulation. We can have a biodegradable polymer, plant-based, for example. So a good example for this is what our MPOB is doing. Okay, our palm oil, because we normally plant palm oils because we want the oil from it, but there's a lot of anti-fruit bunch. So the kind of like normally you see a fruit from palm oil. So what they do is they actually squeeze it out so they get all the oils, and then now you have all the empty bunch without any oil. So what do they do with that? So they part of it, what they can do is either transform that to become a fuel, charcoal, or you can actually process it so that you can get a lot of carbohydrates that you can use for many different purposes. For example, for drug design. Okay, so that one is what we call as biodegradable polymer, so carbohydrates. You can also use non-degradable polymer, for example, polyurethane, is what? Polyacrylate, okay? Or perhaps you can have a copolymer, a mix of those two, or different types of polymer. There's a lot of polymers. I'm sure you guys learn more about polymers than me, or at least in the future. Okay? So liposome, as I mentioned. You can also have a transdermal injection. So instead of having a very big needle, you can have a transdermal injection. So that is an example of different types of technologies related to excipients. So develop this technology with a certain excipients for drug delivery. Okay, so normally it's like this hypodermic needle. So where you can inject it at the very inside of your skin, very close to muscle, for example, and therefore you can feel pain. So these are all pain receptors. So that's why when you being injected using a needle, you do feel pain. So the idea of developing a different technology, for example, a transdermal patch or microneedle, is where you just patch it there to deliver the drug, but you don't feel any pain. Okay, so this technology is being developed in Australia, in the university that I've learned at, but fortunately it's not marketed yet. You can also have inhalable drug delivery methods and excipients. A good example for this asthmatic people inhaling a puffer inhaler. Okay. You can also have polymer coated tablets for oral administration. It's a very very old technology. It's what we normally eat. Okay, when you have all the pills. If you still remember a pill where you can actually open it, have you guys tested that one? So it's basically that. It's a very oldish technology. You can still find it, I guess, once in a while, but most of the time you cannot find it anymore. Alright, so bit of summary, and that's it. Okay, let's move to about 10 minutes. I'm just gonna move to antibiotics. Okay, so far so good. Any question? No? Okay, so make sure you do your assignment. I keep on reminding you guys about your assignment, because so far there's only like one person who actually asked me, because he has a problem. He had a problem installing the program and running it. Nobody else is asking me anything, so I'm hoping that you guys are doing alright. Okay, I don't want you guys to bother me a week before the submission. Saya kata tak boleh install lah, memang kena lah. Okay, saya tak nak jadi macam profesor yang kat UiTM tu, but alright. So again, nak tengok come. So we are now on, what's the date today? day 29 or 31, sorry. Yang on the 31, so this one is week 3. So we're gonna touch a little bit about antibiotics, and then we're gonna continue about antibiotics in the next next week, okay, before moving to analgesic and vaccine, and having your Raya break somewhere in May. Okay, alright. Okay, continuous assessment. This is just an update for you guys. Okay. So we will most likely have a 2-hour exam for your final exam. So it won't be online for this course. And for 2004, it will be as long as the cases does not shoot up again. If it stays like this, then you will have a final exam somewhere, most likely in the exam hall. Okay, because the class is quite big. So okay, dengar tak orang online? Yeah, your exam will be face to face. So wherever you are, come back to Malaysia. If you are somewhere in Sabah, Sarawak, you can probably start planning. Okay, so most likely will be face to face, unless otherwise the directive changes again. Okay, but so far, even me and Dr. Dana and I, we already plan on preparing all your exam questions. Alright, so again, plan ahead. Okay, antibiotics, part 1. So we're going to look at a little bit on the history, classification, example, synthesis, and structure activity relationship. But of course, we don't have enough time to cover everything today. I'm going to continue probably next week, but I'm just going to try and cover whatever we can. Okay, antibiotics. What is an antibiotic? We all know that we consume antibiotics. We take antibiotics when we have a bacterial infection, not viral infection, because viral infection antibiotic does not work in with viruses. Okay, so you only take antibiotics if you are being infected by a bacteria, okay, or fungus. But fungus normally call it antifungus, but antibacterial normally focusing more on microbials. Okay, so please, when you are older, you have a kid and your kid is sick, you brought him or her to a doctor, and then the doctor says no need for antibiotics. Please do not go and ask, but I want antibiotics. Okay, don't do that, because antibiotics is specific for bacteria. It doesn't work if your kid has a flu. A flu is normally a virus, so it doesn't work that way. Okay, so antibiotics is pretty much a secondary metabolite of organism. Mostly is from fungus. Okay, so why is a fungus producing antibiotics? It's well, pretty much it's more on territorial things. Okay, because bacteria and fungus, both of them needs the same source of energy. So one can move. Fungus is like plants, they cannot move. So by producing all these antibiotics, they can actually kill off or stop the bacteria from taking in their resources. Okay, so we call it a secondary metabolite. Why? Primary metabolites is something that we produce for our own body. Secondary metabolites is where we produce, but it's for something else. So in fungus case, it is a secondary metabolite because that metabolite or chemicals is not needed by the fungus itself, but it's actually something that they produce and then they excrete out. Okay? So they produce it outside their body. So so it antagonize growth or survival of other species of organisms. For example, fungus releasing chemicals to inhibit bacterial growth. Okay, so a good example of this is when the discovery of a penicillin. Okay, so if you don't know about that discovery by Alexander Fleming, you just go and read the story a little bit. It's actually quite interesting. Antibiotics are effective at low concentration with selective toxicity. Okay, so these are kind of like the principle or the requirement of antibiotics. Okay. Now, I think the picture over here is just here. So this figure is showing you guys on the phases on how bacterial groups are. So why is this phase important? It's because when we are talking about antibiotics, antibiotics works at a certain phase of a bacterial growth. It doesn't work in all the phases, it works at a certain phase. So for bacteria to grow, so normally there's 4 phases. So lag phase, log phase, stationary phase, and dead phase. So what does it mean? Lag phase is where, for example, you only have like one tiny small bacteria. Okay, so it stays there on its own, doing nothing. It just squirming around here and there, and then at one point, it found the food, the source of food. So the food can be depending on the bacteria. Okay, so some bacteria they like carbohydrate more, some bacteria they like lipids more. So depending on that particular scenario, but if it encounters the food, okay, and especially if the food are in abundance, so what will the bacteria do is they will replicate themselves, so they will grow. So this is what we call as a log phase or exponential phase. It will start to grow unlimited. So it keeps on multiplying until at one point, okay, where some of the bacteria, I would say, tell the other friends that I think we need to stop growing because our food is now scarce, it's very limited. So probably we should stop growing, we should stop multiplicating. So and thus everything, so all the big cluster of the bacteria will stop multiplicating. Okay, so this is what we call as the stationary phase. So it's still there in high abundance, but it no longer producing or replicating. And then normally due to limitation in the resources, so resources can be either light, it can be either food, chemicals, and the other many things. So this one is actually, I learned about this in like 4 hours, but I'm just going to tell you guys in 2 minutes or so. Okay, and then when you have a limited resources, okay, when you have a limited resources, what will happen is now, because there's no more food, they will start to die. Okay, so that's when we go into a dead phase. So just remember these 4 phases, okay, and we will look at why the phases are important in the next few slides. Just remember about these phases. Okay, so these are just to show you guys different classes of antibiotics. So there's so many different types of antibiotics, and these are the dates by which the antibiotics are being discovered. Okay, so beta-lactam is one of the earliest being discovered by Alexander Fleming. It's just before World War One, 1930. Yep. Before World War One, due to accident, we have sulfamides is the first antibiotic that is actually being marketed. So even though beta-lactam was first discovered, it is not the first drug that is being marketed. Why? Because at that point, again, they found it in the lab that penicillin can kill bacteria, but they have problems in trying to deliver the antibiotic for human use. Okay, so at that point, we don't have a lot of technology, so they kind of like try one by one, and in the process of trying, it doesn't work in human for some reason. Okay, but sulfonamides is the first one. It is from more on trial and error thing. So they synthesize it and then test it in a wide range of bacteria, and they found that it's actually good. So they test it again and again and again until at one point they test it in humans and then it works. So this is the first marketed antibiotics. Okay, and then there's few more until at the very end in around early 2000, there's lipopeptides. Okay, so it's a combination of lipids and a peptide as an antibiotic. But up until today, there is no more new antibiotic classes being discovered. Okay, so this is why it's very important for doctors to stop giving you antibiotics if you have a viral infection. Okay, so this is why we're gonna look at what we call as a resistance. So bacterial resistance is very important, because if everybody keeps on consuming antibiotics, the bacteria in our body will try and grow to have a resistance in that particular bacteria. So at one point in time, if you actually get infected, penicillin may no longer work at all. Okay, so you need to go to a different class of antibiotics. So imagine at one point in time in the future, what if bacteria has resistance against all these classes? What will happen to human? You all will die. Okay, so that's why it's very important. Okay, what time is it now? Okay, I think I stopped there. So this is where the log phases are actually important, but We're gonna stop here and continue on next Tuesday. Okay. Last thing from me, make sure you do your assignment. Don't bother me in week 6 above. Otherwise, you can beli your own laptop. Okay, thank you guys. See you around. For those who will be fasting, happy fasting, Ramadan Kareem, and see you guys next week. Thank you.